Phakopsora pachyrhizi is a plant pathogen. It causes Asian soybean rust. Hosts. Phakopsora pachyrhizi is an obligate biotrophic pathogen that causes Asian soybean rust. Phakopsora pachyrhizi is able to affect up to 31 different plant species that belong to 17 different genera under natural conditions. Experiments in laboratories were able to use Phakopsora pachyrhizi to infect 60 more plant species. The main hosts are glycine max, soybean, glycine soya, wild soybean, pachyrhizi xerosis, jicama, *. Preferred hosts. Other hosts were minor or determined experimentally under artificial conditions. Symptoms. The disease forms tan to dark brown or reddish-brown lesions with one to many prominent globe-like orifices. Erdiniospores form from these pores. At initial stages, small yellow spots are formed on the surface of the leaf. These spots may be better observed using assistance of a light source. As the disease progresses, lesions starts to form on the leaves, stems, pod, and petioles. Lesions initially are small, turning from grey to tan or brown as they increase in size and the disease gets more severe. Soon volcano-shaped marks are noticed in the lesions. Disease cycle. Phakopsora pachyrhizi is a fungus which has a spore moved by wind, called erdiniospore. These spores are quite different from others as they don't need an open stomata and natural openings in the leaves. Erdiniospores are able to penetrate the leaf. Pustules are visible after 10 days and they can produce spores for 3 weeks. The disease reaches its climax when the crop begins flowering. The cycle of the pathogen continues until the crop is defoliated or until the environment becomes unfavorable to the pathogen. The Asian soybean rust is a polycyclic disease which within the disease cycle the asexual erdiniospores are responsible to keep infecting the same plant. Teleospores, sexual spores, are the survival spores that overwinter in the soil. Basidiospores are the spores that are able to contaminate an alternative host. The erdiniospores need a minimum of 6 hours to infect leaves at a favorable temperature, between 15 to 24 degrees Celsius. Environment. The favorable conditions for the disease to progress are related to temperature, humidity, and wind. The appropriate temperature for the pathogen to be active is from the range of 12 to 29 degrees Celsius. More efficient between 18 to 26.5 degrees Celsius. Aside the temperature, the humidity must be really high, about 90% or more for more than 12 hours. A significant amount of wind is also important for the pathogen to move from a plant to the other. Current in the United States. Infected plant can be found in Florida, Georgia, Louisiana, and Texas. Risk factors. Uridospores are wind-blown and are produced abundantly on the infected tissue of soybeans or other legume hosts. Management. The disease is often controlled using the fungicides oxycarboxyn, trifluorine, or triclopyr. Phakopsora pachyrhizi is a pathogen that acts quite fast in contaminating the host. The plant can be severely contaminated in a short period as 10 days. This makes difficult to control the disease, as it does not just spread quickly, but the progression of the disease is also fast. That is why it is important to implement control techniques as soon as possible. Genetic resistance. The disease may be controlled by using genetic resistance, but it has not exhibit great results. It is not effective after a certain period. Chemical control. A second management that can work is using fungicides, but it is only efficient at early stages of the disease. The disease spreads fast and it is complicated to control after certain stages. So it is important to act careful around contaminated plants as the spores can be attached to clothing and other materials and infect other plants. Index Fungorum. USDA's fungal database.